transplanted into a woman. Or they may be donated for research purposes with informed consent of the donors. The zygote, after 4 to 6 days of fertilization, becomes a mass of cells called a blastocyst. The blastocyst consists of an inner cell mass and an outer cell mass. The outer cell mass becomes part of the placenta, and the inner cell mass differentiate to become all the structures of an adult organism. This mass is the source of embryonic stem cells called totipotent cells. When extracting embryonic stem cells, the blastocyst stage signals when to isolate stem cells by placing the inner cell mass of the blastocyst into a culture dish containing a nutrient-rich broth. In the absence of stimulation to differentiate, they begin to divide and replicate while maintaining their ability to become any cell type in the human body. Eventually, these undifferentiated cells can be stimulated to create specialized cells at various points during the process of generating embryonic stem cell lines. Scientists perform characterization to see the viability of these cells. They exist through the body after embryonic development and are found in tissue and organs including brain, bone marrow, peripheral blood, blood vessels, skeletal muscle, skin, teeth, heart, gut, liver, ovarian epithelium, and testis. They are thought to reside in a specific area of each tissue called a stem cell niche. They can differentiate into cells of same lineage. The adult stem cell can renew itself indefinitely and can differentiate to yield some or all of the major specialized cell types of the tissue or organ. The primary roles of adult stem cells in a living organism are to maintain and repair the tissue in which they are found. Stem cells may remain present for long periods of time until they are activated by a normal need for more cells to maintain tissues or by disease or tissue injury. Scientists have found adult stem cells in many more tissues than expected, like in brain and heart. This finding has led researchers and clinicians to consider use of adult stem cells for transplants. If the differentiation of adult stem cells can be controlled in the laboratory, these cells may become the basis of transplantation-based therapies. Now, let us see the similarities and differences between human embryonic and adult stem cells. Both have advantage and disadvantage regarding potential use of cell-based regenerative therapies. One major difference between adult and embryonic stem cells is their different abilities in the number and type of differentiated cell types they can become. Embryonic cells can become all cell types of the body because they are pluripotent, while adult stem cells are thought to be limiting to differentiating into different cell types of their tissue of origin. The embryonic cells can be grown relatively easily in culture, while adult stem cells are rare in mature tissues, so isolating these tissues from an adult tissue is challenging. The adult stem cells and tissues derived from them are less likely to initiate rejection after transplantation by the immune system. This is because a patient's own cell could be expanded in culture, coaxed into assuming a specific cell type, and then reintroduced into the patient. However, embryonic stem cells may initiate rejection. Differentiation. Stem cells can differentiate into hematopoietic stem cells, which give rise to all types of blood cells: RBCs, lymphocytes, natural killer cells, granulocytes, monocytes, and macrophages. Mesenchymal stem cells that have been reported to be present in many tissues, those from bone marrow stromal stem cells, skeletal stem cells, give rise to a variety of cell types like bone cells and cartilage cells, fat cells, and stromal cells that support blood formation. Neural stem cells in the brain give rise to nerve cells and non-neuronal cells like astrocytes and oligodendrocytes. Epithelial stem cells in the lining of the digestive tract, it occurs in the deep crypts, giving rise to several cell types like absorptive cells, goblet cells, and enteroendocrine cells. Skin stem cells. They occur in the basal layer of the epidermis and at the base of hair follicles. The epidermal stem cells give rise to keratinocytes, which form a protective layer. The follicular stem cells give rise to both the hair follicle and epidermis. Transdifferentiation. Many experiments have reported that certain adult stem cell can differentiate into other cell types outside the cell's predicted lineage. For example, the brain stem cells differentiate into blood cells or blood-forming cells, differentiate into cardiac muscle cells, etc. by the process called transdifferentiation. Scientists demonstrated that this may be because of well-controlled process of genetic modification. This strategy may help to reprogram available cells into other cell types that have been lost or damaged due to disease. For example, if the pancreatic beta cells that produces insulin are lost or damaged in diabetes, it could possibly be created by reprogramming other pancreatic cells that can secrete insulin. Adult somatic cells can be reprogrammed to become embryonic stem cells and are known as induced pluripotent stem cells or iPSCs. Thus, a source of cells can be generated that are specific to the donor, thereby increasing the chance of compatibility during tissue regeneration. Growing cells in the laboratory is known as cell culture. Stem cells are either extracted from adult tissue or from a dividing zygote in a culture dish. Once extracted, scientists place the cells in a controlled culture that prohibits them from further specializing or differentiating further, but allows them to divide and replicate. The process of growing large numbers of embryonic stem cells has been easier than growing large numbers of adult stem cells, but progress is being made for both cell types. Tissue collection and transportation. The first and most important element in the collection of tissue is the cooperation and collaboration of the clinical staff, such as the surgical, one of the nursing staff, or pathologist. The tissue should be collected in a double-wrapped, sealed polythene bag or container and an outer padded envelope provided with the name, address, and phone number of the recipient at the laboratory. The label should also state clearly, "Do not freeze." Refrigeration during transport is not usually necessary unless the delivery takes more than an hour or two, which can be done with refrigeration packs. The tissue will be used to corroborate the origin of any cell line that is derived from the sample by DNA profiling. Biosafety and ethics. All procedures involved in the collection of human material for cell culture must be passed by the relevant hospital ethics committee. A form will be required for the patient to sign authorizing research use of the tissue and preferably disclaiming any ownership of any materials derived from the tissue. All human material should be regarded as potentially infected and treated with caution. Each laboratory will have its own biosafety regulations that should be adhered to. Record keeping. When the samples arrive at the laboratory, it should be entered into a record system and assigned a number. The record should contain the details of the donor, tissue site, collection medium, time in transit, treatment on arrival, primary desegregation, and culture details, etc. The embryonic stem cells, called HESC, are generated by transferring cells from a pre-implantation stage embryo into a plastic laboratory culture dish coated with mouse embryonic skin cells, specially treated not to divide. This coating layer of cells is called a feeder cell or feeder layer. The culture dish contains a nutrient broth known as culture medium. The cells divide and spread over the surface of the dish. The mouse cells in the bottom of the culture dish provide the cells a sticky surface to which they can attach. Also, the feeder cells release nutrients into the culture medium. Researchers have now devised ways to grow embryonic stem cells without mouse feeder cells. This is a significant scientific advance because of the risk involved, where viruses or other macromolecules in the mouse cells get transmitted to the human cells. If the plated cells survive, divide, and multiply enough to crowd the dish, they are removed gently and plated into several fresh culture dishes. The process of replating or subculturing the cells is repeated many times for many months. Each cycle of subculturing the cell is referred to as passage. Once the cell line is established, the original cells yield millions of embryonic stem cells. At any stage in the process, batches of cells can be frozen and shipped to other laboratories for further culture and experimentation. Some examples of potential treatments include regenerating bone using cells derived from bone marrow stroma, developing insulin-producing cells for type 1 diabetes, and repairing damaged heart muscles following a heart attack with cardiac muscle cells. Once stem cells have been allowed to divide and propagate in a controlled culture, the collection of healthy, dividing, and undifferentiated cells is called a stem cell line. Once under control, the stem cells can be stimulated to specialize as directed by a researcher, a process known as directed differentiation. Embryonic stem cells are pluripotent and are able to differentiate into more cell types than adult stem cells. Stem cells are categorized by their potential to differentiate into other types of cells. Embryonic stem cells are the most potent since they must become every type of cell in the body. To confirm experimentally that a putative adult stem cell is indeed a stem cell, scientists tend to show either that the cell can give rise to these genetically identical cells in culture and or that a purified population of these candidate stem cells can repopulate or reform the tissue after transplant into an animal. Laboratories that grow human embryonic stem cells use several kinds of tests, including growing and subculturing the stem cells for many months to ensure that the cells are capable of long-term growth and self-renewal. The cells must be healthy and undifferentiated. Using transcription factors like OCT4 and NANOG, which are used to maintain the stem cells in an undifferentiated state, capable of self-renewal. These help turn genes on and off at the right time to ensure cell differentiation and embryonic development. Use of specific techniques to determine the presence of particular cell surface markers typically produced by undifferentiated cells. Testing whether the human embryonic stem cells are pluripotent by, number 1, allowing the cells to differentiate spontaneously in cell culture, number 2, manipulating the cells so they will differentiate to form cells characteristic of the 3 germ layers, number 3, injecting the cells into a mouse with a suppressed immune system to test for the formation of a benign tumor called a teratoma. Although complete agreement among scientists as to how to identify stem cells are not prevalent, yet most tests are based on making sure that stem cells are undifferentiated and capable of self-renewal. Preservation of cells for a long time are done by freezing secure stocks to be maintained without aging and protect them from problems of contamination, incubator failure, or medium and serum crisis. Ideally, 10 to the power 6 to 10 to the power 7 cells should be frozen in 10 ampoules in glycerol or dimethyl sulfoxide at 5 to 10%. Smaller stocks can also be used as per requirement and availability. The normal procedure is to freeze a token stock from the surplus cells and expand remaining cultures to confirm the identity of the cells and absence of contamination. Next, a seed stock of 10 to 20 ampoules are frozen down. 1 ampule thawed from this stock can then be used to generate a using stock. Cross-contamination. This is less of a problem with short-term cultures, but the risk remains that if there are other cell lines in use in the laboratory, they can cross-contaminate even a primary culture, or misidentification can arise during subculture or recovery from the freezer. Hence, precautions must be taken to avoid cross-contamination, namely by not handling more than one cell line at a time, not having culture vessels and medium bottles for more than one cell line open at one time, by avoiding the use of same pipette or other device for different cell lines, by not sharing media or other reagents among different cell lines or with other people, by ensuring disinfecting with 70% alcohol during any spillage immediately, by retaining a tissue or blood sample from each donor and confirm the identity of each cell line by DNA profiling during freezing seed stock or before the cell lines is used for experimental work or transplantation, by keeping photographs of each cell line at low and high densities and consult this regularly when examining cells during maintenance. To avoid microbial contamination, antibiotics are often used during collection, transportation, and dissection of biopsy samples because of the intrinsic contamination risk of these operations. If the culture grows well, then antibiotics can be removed from the bulk of the stocks at first subculture, retaining one culture in antibiotics as a precaution if necessary. Antibiotics can lead to lax aseptic technique, they can inhibit some eukaryotic cellular processes, and also hide the presence of a microbial contamination. If a culture is contaminated, the culture should be discarded before it can spread the contamination to other cultures. Most bacterial, fungal, and yeast infections are readily detected by regular, careful examination with the naked eye, for example by a change in the color of the culture medium and on a microscope. There are many ways in which human stem cells can be used in research and the clinic. A more complete understanding of the genetic and molecular controls of these processes may yield information about how such diseases arise and suggest new strategies for therapy. Stem cell therapy uses stem cells to treat or prevent a disease or condition. Bone marrow transplant is a crude form of stem cell therapy that has been widely used clinically for many years without controversy. Research is underway to develop various sources for stem cells and apply stem cell treatments and research. Stem cells help in basic research such as clarification of complex events that occur during human development and understanding molecular bases of cancer. Molecular mechanisms for gene control, signals in gene expression and differentiation. The undifferentiated stem cells eventually differentiate partly because of a particular gene is turned on or off. Stem cell researchers may help to clarify the role that genes play in determining what genetic traits or mutations we receive. Understanding the theory of cancer. Stem cells help in cell-based therapies such as regenerative therapy to treat Parkinson's, Alzheimer's, spinal cord injury, stroke, severe burns, heart disease, diabetes, osteoarthritis, wound healing, and rheumatoid arthritis. The learning disability due to congenital disorder, anti-cancer treatments, repair hearing and restore vision, and repair damage to the cornea and amyotrophic lateral sclerosis or ALS. Organ and tissue regeneration is probably the most important possible application of stem cell research. Stem cells could potentially be used to grow a particular type of tissue or organ. Stem cells that lie just beneath the skin, for example, have been used to engineer new skin tissue that can be grafted onto burn victims. Healthy heart cells developed in a laboratory may be transplanted into patients with chronic heart disease, repopulating the heart with healthy tissue. Similarly, type 1 diabetes can be cured by directing the differentiation of human embryonic stem cells in cell culture to form insulin-producing cells that can be eventually used in transplantation therapy. Adult hemopoietic stem cells found in bone marrow and blood have been used for years to treat blood diseases such as leukemia, sickle cell anemia, and other immunodeficiencies. These cells are capable of producing all blood cell types such as RBCs that carry oxygen to white blood cells that fight disease. The cells are extracted from bone marrow or umbilical cord and placenta. Stem cells help in biotechnology such as in drug discovery and development by providing specific cell types to test new drugs. In screening of potential drugs, human stem cells are currently being used to test new drugs. New medications are tested for safety on differentiated cells generated from human pluripotent cell lines. Cancer cell lines are already being used to screen potential anti-tumor drugs. The availability of pluripotent stem cells would allow drug testing in a wider range of cell types to reduce animal testing. Harvesting of embryonic stem cells in order to avoid the problem of immune rejection, scientists are experimenting with different research strategies to generate tissues that will not be rejected. One of the disadvantages of stem cell treatments is that it may require immunosuppressant because the patient may receive radiation before the transplant or because the patient's immune system may target the stem cells. It is therefore preferable to use stem cells from the same patient who is being treated. Pluripotency in certain stem cells could also make it difficult to obtain a specific cell type. It is also difficult to obtain the exact cell type needed because not all cells in a population differentiate uniformly. Undifferentiated cells can create tissues other than desired types. Some stem cells form tumors after transplantation in embryonic stem cells. Fetal proper stem cells induced pluripotent stem cells. Fetal proper stem cells form tumors despite multipotency. Scientists and researchers are interested in stem cell cultures for several reasons. Stem cells extracted can be induced to become any desired cell type. This property makes them powerful enough to regenerate damaged tissues under the right conditions. However, the use of stem cell is still under controversy. Stem cell research helps to identify how undifferentiated stem cells become the differentiated cells and understand about the complex events that occur during human development. A more complete understanding of the genetic and molecular controls of these processes may yield information about how such diseases arise and suggest new strategies for therapy. Stem cell therapy uses stem cells to treat or prevent a disease or condition. To summarize, stem cells offer exciting promise for future therapies, but significant technical hurdles remain that will only be dealt with through years of intensive research. In the coming years, with increasing ability of scientists to isolate and culture embryonic stem cells and grow induced pluripotent stem cells, we can hopefully reach the zenith of treatment of diseases.
Hello viewers, welcome to this particular lecture on malarial parasite and its control. Malarial parasite is represented by a species or genus Plasmodium belonging to Protozoa. This particular parasite is actually developing in human, but it completes the life cycle in mosquito, that is female Anopheles mosquito. So in this particular lecture, I shall be considering the malarial parasite, the different types of malarial parasite, the life cycle of malarial parasite and its historical development, and also I shall be discussing the different damages that this parasite causes and the specific ways by which this disease can be controlled. So malarial parasite actually, mainly the common malaria is the Plasmodium vivax, which is the normal malaria or benign malaria. There may be also the Plasmodium malari, which is also called the quartan malaria. There may be also the Plasmodium ovale, which is called the Tashian malaria. And apart from these three, there may be the dangerous Plasmodium falciparum, which is also called the malignant malaria, and also the Plasmodium knowlesi, which is a zoonotic disease, mainly invading the animals, And lastly, there is another type, which is the Plasmodium hyoli, which is African malaria. Now, all these particular figures, you can see, this is the parasite or the merozoite of the parasite, Plasmodium, and these are the RBC. So all these photographs are taken in a particular blood sample. So the best way to detect malarial parasite is testing of blood. And apparently, this particular parasite can grow both outside the RBC, but mainly it will feed on the RBC, so it gets inside and it will produce different excretory matter inside the RBC by feeding on the hemoglobin. So basically, it is a blood parasite which is surviving inside RBC for a long time, and also it can survive in liver. And apparently, by seeing the different pattern of growth or stages of growth, we can actually identify it. So a very good histologist, just by seeing the infected blood slide, can tell you what is the nature of the pathogen, whether it is Plasmodium vivax or it is Plasmodium falciparum or any other species, right? This is a comparison of the different stages of malarial parasite, and that actually can be understood by the way they grow inside the RBC. Coming to the systematic position, as I told you, it is a protozoan pathogen, but nowadays it is also called Apicomplexa. It belongs to the class of Arconidiacida, and also it is having the order of Hemosporida. It is having the family of Plasmodidae, and the genus is Plasmodium, and the species may be Vivax or Falciparum or others. Now coming to the historical development of this particular disease. This disease actually was reported for the first time in 2700 BC in China, and not only that, people at that time could actually treat this type of fever with some hemp bark extract. So they knew the art of treating malaria with some hemp bark extract, which perhaps was some anti-malarial alkaloid. Much later, maybe that also, maybe in 500 or to 500 to 1200 BC, that was the time period, there was a severe infection all over the Roman Empire. It spread across Italy, Sardinia, and it was growing along the Tiber River. And so much dangerous was the fever, it was actually causing a lot of death. And it was called, or it became famous in the ancient Rome as Roman fever, and still it is important in that name. Okay? Now, it was Charles Laveron in the year 1880. This person could for the first time discover that malarial parasite is surviving in human blood. And this particular discovery actually made him win the Nobel Prize in 1907, much later. And it was Finlay who actually was thinking that this particular disease was having some association with mosquito. And he wrote a paper in 1881 stating that Anopheles mosquito is involved in it, but he couldn't prove it though. But it was this person who is Ronald Ross, starting from the year 1894 till 1898, he worked in PG Hospital, Kolkata, and also in other parts of the country, and exclusively he proved that the female Anopheles mosquito requires human blood for the maturation of the egg, and by that way they transmit the malarial parasite to human beings. So in that way, human and mosquito, these are the two hosts of the malarial parasite, Plasmodium, where definitely he proved that the sexual cycle occurs inside the mosquito. So this particular disease or this particular pathogen is having a definitive host in the form of female Anopheles mosquito, and the alternate host where the asexual reproduction occurs takes place in human, and that made him win the Nobel Prize in the year 1902. Now that was in 1898 this discovery was made, and it was Walter Reed who in the year 1900 he confirmed the findings of Ross. And much later, this is a something different discovery in the period between 1917 to 1940. This person, that is Julius Wagner Jauregg, he showed that this particular parasitic infection can prevent a bacterial infection. So if a person suffering from syphilis was inoculated with malarial parasite, the disease syphilis could be controlled, because at that time you understand there was no suitable reply to syphilis infection. But definitely that was not continued though, because you know, for treating one disease, you cannot make a person suffer from another disease. Okay, this particular disease is growing worldwide, but you can clearly see it is only growing along the tropics. So it is a tropical problem. It is not a problem in the temperate climate. So it is growing all over Southern America, it is growing in Africa, and of course India. But the most important area where it is endemic and causing havoc is Africa. Number of death also is higher. And perhaps that is the reason also that Ronald Ross, in order to do research on Plasmodium, had to come to a tropical country, and that is India. So malarial parasite is invading multiple organs. Different organs are invaded. It can be symptoms like headache, it can have muscular fatigue, back pain, there may be chills and sweating, there may be dry cough, there may be enlargement of spleen, and there may be stomach nausea or vomiting. And at the same time, there may be sometimes some complication, and this complication may be acute or chronic. The acute one may be in the form of cerebral malaria invading the brain, or the chronic may be in the form of anemia, placental malaria, or severe jaundice. Okay?
Sir, why is jaundice associated with malaria?
You see, malaria, I told you, is growing on RBC, so the RBC is destroyed, so hemoglobin is destroyed, and hemoglobin will produce bilirubin, so the serum bilirubin concentration goes up, which is equivalent to jaundice, because the more number of RBC will be destroyed, there will be more bilirubin. So you understood? Good. Now coming to the life cycle of Plasmodium vivax. So as I told you, clearly you can see this life cycle is having the human cycle and the mosquito cycle. This human cycle, the mosquito is biting the human containing these particular sporozoites, which will enter the liver, and this cycle is called exoerythrocytic cycle or pre-erythrocytic cycle. What is the principle? This particular malarial parasite will undergo asexual division, producing the sisons, and these sisons will enter into the RBC and feed on the hemoglobin, produce a huge vacuole, we call it signet ring, and ultimately towards the end of this erythrocytic cycle, it will produce the gametocytes. And these gametocytes will be transmitted again to mosquito, where they will undergo sexual reproduction, and they will finally produce the oocyst, and again they will be undergoing reduction division to produce the sporozoites. So you can see that though the malarial parasite is spending more time in human, but yet mosquito, where the sexual reproduction occurs, is actually the definitive host. It is not the human. Human is alternate host. Similarly, when we consider the life cycle of Plasmodium falciparum, you can also see that this infection is more or less the same. There will be the liver cycle, there will be the RBC cycle, there will be the gametocytes produced, and finally it will be in the mosquito where they are growing. But the question is, sometimes this particular parasite will cross the blood-brain barrier, will cause the infection, creating a fatal condition. So this Plasmodium falciparum or malignant malaria, it is so called because the way it is dividing, faster division, is also called cerebral malaria. This particular erythrocytic is being killed by that Plasmodium, and this is producing a huge particular vacuole. And this vacuole is not the part of the cell, but this vacuole is actually the part of the Plasmodium parasite, and this is called the signet ring. So an efficient histologist, just by seeing the pattern of the signet ring or the efficiency of the signet ring, can actually say which particular Plasmodium has attacked that person. As I told you, that this particular infection is mainly in the blood. So what is happening? Ultimately ultimately the malarial parasite is feeding on blood. So they are destroying the different components of globin and also the heme. So what do? They say, for example, the globin fragments are converted to smaller peptides, and these peptides are ultimately utilized as amino acids. And these amino acids will be utilized by the mosquito, and at the same time, there will be the heme protein. The heme protein will on one side produce the heme, and on the other side, it will produce peptides. At the same time, it produces a waste product. So this particular waste product, hemozoin, is deposited in the particular blood, and that hemozoin is toxic for us, so the body temperature goes up, and that results in chill and fever in humans.
Sir, I've heard there is recurrent fever in case of malaria. Why exactly is that?
You can understand this particular hemozoin production cannot occur once. It will occur, say, 3-4 cycles are there, and the moment the hemozoin is produced, then again the temperature will go up, the chill will be there, and it will be destroyed. And again the malaria cycle will continue. They will again invade fresh RBC, and this fresh set of RBC will again produce the hemozoin, and that will take some 1 or 2 days time. And that is the reason why this particular malarial parasite is classified on the basis of tertian, quatern, or other varieties. Because the chill comes or fever comes, say, every 24 hours, every 48 hours, every 72 hours. This is because of this hemozoin cycle. Okay, so it is a cyclopropagating parasite. Clear to you? Good. Now, as I was telling, that in the life cycle, it is not really happy in the blood. It has to go somewhere else. It is adventurous. So this particular malarial parasite will get into the brain. It will cross this particular blood-brain barrier. What is blood-brain barrier? It is the barrier between the CSF and the blood. And you know this area is formed of astrocyte. So they will cross this particular blood-brain barrier and get into the brain. And the moment it gets into the brain, it needs huge concentration. So there is some strategy. What is that? They produce some specific protein EMP1, which will release the GPI anchored protein, and they will finally produce the adenosine. So the adenosine will help that particular parasite to adhere to the wall of the blood vessel. So that means, in fact, they will not be carried away. So the huge number of parasite concentration there is in the brain capillaries. And the moment they enter into the brain, they will cause serious infection, hypersensitivity, and that particular symptoms can be fatal. The person can get into coma, and that is cerebral malaria.
Sir, what is the mechanism of this malarial encephalopathy?
You see, encephalon is the other name of the brain. Because of the infection, there is some typical pathogenic symptoms developing, and this encephalopathy actually indicates swelling of this particular brain and the irregularity within the CSF, which also is accompanied by cytokine production, hypersensitivity, and ultimately resulting in death. So the basic mechanism is initiated by the crossing of the malarial parasite from blood to the brain. Now, apart from brain, I was telling you that this liver is a very important organ where the malarial parasite will reside. It is a repository for the malarial parasite, and this particular liver is showing enlargement, and the malarial trophozoites or sporozoites may remain in liver, may reproduce in liver, and they might develop conditions similar to jaundice or hepatitis, or even sometimes liver cancer. So that way it is really dangerous that this particular liver which is a very delicate organ, which is the metabolic king of our body. All the metabolic activities takes place within liver, but this particular organ is getting affected because of malarial parasite, and our digestive power also is reduced. Now, there are some people who are showing resistance to this malarial parasite. Who are they? People who are resistant may include the person suffering from sickle cell anemia. And this is how these blood vessels or blood cells will look like. That is, they are looking like a sickle. Instead of this round RBC, you are having sickle-shaped RBC. Why this occurs? Because you know this is a normal hemoglobin having 2 alpha, 2 beta chain. And in case of sickle cell anemia, you can see this is the deformity in the beta chain. Why this deformity occur? It is because of the 6th position glutamic acid replaced by valine. So this is a point mutation that brings about sickle cell anemia. They are protected. Why protected? Because that particular sickle-shaped cell is not allowing or accommodating the parasite inside. But they are not lucky enough, because anyway they are suffering from the severe anemia and they are having limited number of days in their life. But this type of persons who are producing G6PD, they are normal. They are having this particular malarial infection where glucose is converted to phosphogluconate. But there are certain people who are producing G6PD deficient or who are not producing G6PD. These people are also resistant. Why? Because normally the ROS or reactive oxygen species are not removed or sequestered. So because of this ROS, this particular malarial parasite can be actually checked. And there is another group. This is again something very interesting. There is a particular Duffy protein receptor, and this Duffy protein receptor is the gateway for the malarial parasite to come in. This receptor, if it is not developed or if it is modified, mutated in any way, then the malarial parasite cannot enter the RBC. And I told you, if it cannot enter the RBC, it will have to fast, because its food is hemoglobin. So they cannot feed on hemoglobin, so they die, they perish. So even if the parasite is inside, but because of the absence of the Duffy protein, they cannot go in and they die. Okay? So these people are also resistant. There maybe I was telling that malaria can be detected in different ways. It is because of the antibodies. So what is the mechanism? You see, the moment these B lymphocytes are active, this B lymphocyte will have the merozoite protein or merozoite antigen. They will activate a particular HLA2, and this HLA2 will bind with this merozoite protein. So if we can measure the amount of HLA2, what is the amount of this particular antigen with a specific antibody, we can easily detect, or we can easily state that in what way we can detect malaria. So one way of detecting malaria is the antigen-antibody reaction between the HLA2 and the CD4, and this HLA2 is combining with the merozoite protein. And ultimately, when this particular B cell will be having this merozoite, it will remain there for some time, but not for a long time. It will have to come out in blood and invade another RBC and feed on them. Ultimately, they have to go to the RBC. Only temporarily they can remain in the WBC, and when they are inside the WBC, you can detect them by this antigen-antibody mechanism. And this mechanism, you can see clearly, this is a positive result because these antibodies are there, this binding with a specific antigen, and you can say that this person is having malarial parasite in his blood. So this mechanism is called rapid detection test or rapid immunological test for detecting malaria. But of course, I told you, everywhere you may not have this facility of RDT. So what you do is, you will check the slide. And checking the slide, you can actually stain this malarial parasite, and you can differentiate between the structures of Plasmodium falciparum, Plasmodium ovale, or Plasmodium vivax, or Plasmodium malari. And if he is a very experienced pathologist, he will immediately tell you what is the type of pathogen that has entered your blood. Now we come to a very important aspects of preventing this malarial disease. Ideally, we should prevent malarial disease by checking the pathogen through their vector. So you prevent mosquito. So what should be the ideal way? Mosquito net. You can use mosquito repellent. You can use closed window panes so that mosquito cannot go in. Because most of the time, let me tell you, they are in the evening, they are coming inside your house and they are feeding on the blood. Because all through the night they will metabolize the blood, and you can see very often mosquito, their tummy is full with blood and they are actually resting in a place. And at that time they are utilizing that protein. So if you can prevent that, ultimately it will be reduced. The malarial risk will be reduced. And there can be pyrethrin, which is actually an insecticide that kills mosquito from pyrethrum flower. So this is a chemical control. And apart from that, there may be physical control, cultural control. What is cultural control? You will will not allow the mosquito breeding places. You will not allow accumulation of water. What is physical control? You will make a barrier. It can be mosquito net, or it can be any sort of layer which will prevent the entry of mosquito or biting of the mosquito. And similarly, you can also have awareness. Many people doesn't know. If I ask you how malaria is caused, many people will say that it is caused by mosquito. But actually they don't know it is caused by the plasmodium inside the mosquito. So this particular phenomenon has to be taught to other people. And this is also the duty of the government. And they may make people aware by observing the National Malaria Month, which is the month of June. Why month of June? Because more rain, more water, more malaria. Right? Now you see, structure of quinine. Quinine comes from cinchona plant. Quinine is not the only drug. If I ask you how is malaria controlled, you will say controlling vector. But if the mosquito has already bitten and already there is infection, then you have to take drug. But without testing blood, if you take a drug, naturally it is not doing good to you because it will destroy the liver cells. You will suffer from disorder of liver. So you have to test the blood. You have to be rest assured you have malaria. Then only you take quinine. And these drugs are of this type: chloroquine, primaquine. And sometimes, this is very important, sometimes people already take it as a prophylactic measure. They don't have malaria, but they take this drug so that malaria doesn't take place. So these are one group of drugs which are quinolones. There may be sesquiterpenes, there may be naptoquinone. So artemisinin, quinine, and atovaquone, these are the main drugs used against malaria. And there is a typical regimen, meaning, say for example, if you use the same drug for a continuous period, that drug will be of no use. So normally the regimen is like this: you start with chloroquine. If chloroquine is not there, you get into primaquine. If primaquine is not there, you get into metacalfine. Metacalfine is not there, you get into other drugs like artemisinin or artemisinin derivatives. And some of these drugs actually are toxic. So you cannot take a toxic molecule for a prolonged period because that will be having a negative effect on your liver, on your general health. Because liver is the metabolic king, I told you. So this is a very important drug, the benzalmenadione, which is preventing both. It is preventing the sporozoite development in the blood, and at the same time, it is inhibiting the hemozyme formation, or it is inhibiting the metabolism of hemoglobin. So in both ways, the malarial parasite is in a fix and it perishes. Now we come to a very important aspect, which is that the aspect of artemisinin resistance. Whenever we have a particular drug, a mosquito or it is, parasite will develop resistance. So that drug cannot percolate inside. So what to do then? Naturally, we have to prevent the disease. Whether we should go for vaccination or not to prevent the disease. Now what can be the possible way? The possible way is we take, this is the malarial parasite, Plasmodium falciparum, from where we are taking the specific surface protein, we are inciting it into a virus. This is a virus body, and this virus body is inserted into the normal cell where they are producing this huge amount of malarial vaccine protein. And this virus is non-harmful to human. So this is the basis by which we produce a particular antibody against this known Plasmodium falciparum protein, right? And naturally, whenever there will be a new antigen, that particular antibody will prevent its replication. So that way the vaccine can be a very effective means, but that always this vaccine may not work. Now why it doesn't work? You must understand, basically this is binding of a sporozoid with a gateway protein. That is the point through which it will enter. And if the gateway protein is somewhere is blocked, then the virus cannot enter and it will perish. So this is the working principle of the malarial parasite. But the question is, this parasite is having multiple stages. It is having the sporozoid, it is having the hypnozoid, it is having the cryptozoid, it is having the merozoid. All these stages are different. And not only that, replication rate is also faster. So there is a problem of the titer. If the titer is not enough, malarial parasite is replicating faster, then it will overgrow. At the same time, sometimes new protein is there, you have answer to one protein, but if some other protein is there, it will enter. You cannot prevent it. So that is why the efficacy of the vaccine is questionable. But still, research is on, and there will be some vaccine which will be effective in the long run, of course, in future. So I shall conclude the control strategy. At the outset, we should control mosquito. We will prevent their breeding. We take a pledge that we will not allow water accumulation anywhere, because that water is the breeding place of mosquito. We can have biological control by, say, by using some anti-mosquito protein in the form of Bacillus sphaericus toxin. We will kill that mosquito larvae. We can also have physical barrier, curtain or net by which we will prevent the mosquito entry. We can use chemicals by which mosquito biting is not possible. And at the same time, even if after all these things we are suffering, we get into the regimen of using chemical drugs, and these drugs we don't continue the same drug. We will vary the drug. Initially we take chloroquine and primaquine. Then if it is not working, we go to artemisinin. So in this way, we do not really allow that particular malarial parasite to be exposed to a compound, because you know when a particular malarial parasite is entering our body, it is residing for quite some time within the liver, and that is the reason why if you are suffering from malaria, you will not be allowed to donate blood for the next 6 months, because even if the symptoms are not there, you never know the malarial parasite is very nicely located in your liver. So students, I have discussed the different aspects of the Plasmodium, that is the malarial parasite. I have discussed the historical aspects of discovery. I have discussed what are their life cycle, in what way they actually cause damage to different organs. I have also discussed the life cycle stages and in what way they are propagating. And finally, I have discussed the different means by which we can control the disease. Thank you.
In this session, let us discuss the properties of proteins, one of the most important biomacromolecules. Proteins have many physical, chemical, mechanical, and biological properties suitable for their roles in biochemistry. Let us now look at the physical properties of proteins. Major physical properties of proteins are highlighted here. Proteins are usually colorless and tasteless, homogeneous and crystalline. They range in shape from simple crystalloid spherical structures to long fibrillar structures. Two distinct shapes have been characterized. Globular proteins, they are usually spherical in shape. Examples include pepsin, intestine, insulin, and ribonuclease, etc. Fibrillar proteins, these are thread-like or ellipsoidal in shape. Most of the structural studies of proteins have been done using these fibrillar proteins. Examples of such proteins include fibrinogen, myosin, etc. Proteins generally are very high molecular weights ranging between 5 kilodaltons and 1000 kilodaltons. Due to their giant sizes, proteins exhibit many colloidal properties such as the following. They have extremely low diffusion rates. They may cause considerable light scattering in solution, resulting in visible turbidity. This is known as Tyndall effect. Denaturation is a process that involves the disruption of the secondary and tertiary structures of the protein, leading to the loss of biological activity. Often, denaturation is followed by coagulation, a process in which denatured protein molecules form large aggregates which precipitate from solution. Denaturation may be achieved by a variety of agents, both physical and chemical. Physical agents include mechanical action, such as shaking, heat treatment, cooling and freezing operations, high hydrostatic pressures, rubbing, UV rays, and ionizing radiation such as X-rays, radioactive and ultrasonic radiations, etc. Let us have a quick glance at the mechanisms by which various agents cause denaturation. Proteins are amphoteric in nature, just like amino acids. They act as both acids and alkalis. Proteins migrate in an electric field, and the direction of migration depends upon the net charge carried by the molecule, which is influenced by the pH value. Ion binding capacity. Being amphoteric, proteins can form salts with both anions and cations. A mixture of proteins at a given pH will include both cations and anions, and salts of protein-protein combinations will be formed. The solubility of a protein is greatly influenced by pH. Solubility is lowest at the isoelectric point, that is pI, and increases with increasing acidity or alkalinity. This is because when the protein molecules exist as either cations or anions, repulsive forces between ions are high, as all the molecules possess excess charges of the same sign, positive or negative. Thus, they will be more soluble than when they are at the isoelectric point. Optical activity. All protein solutions rotate the plane of polarized light to the left, that is, proteins are levorotatory. Now discuss the major chemical properties of proteins. Hydrolysis. Proteins are hydrolyzed by a variety of hydrolytic agents. Hydrolysis by acidic agents. Proteins, upon hydrolysis with concentrated hydrochloric acid, yield amino acids in the form of their hydrochlorides. Hydrolysis by alkaline agents. Proteins may be hydrolyzed with alkalis such as sodium hydroxide, or proteins may be hydrolyzed by hydrolytic enzymes, that is, proteolytic enzymes. Under mild conditions, certain enzymes such as pepsin and trypsin can hydrolyze the proteins, and ultimately they may be reduced to their constituent amino acids. Let us now look at the reactions involving carboxylic groups present in proteins. Reaction with alkalis, which is known as salt formation. The carboxylic groups of amino acids present in proteins can release protons, resulting in the formation of carboxylate ions, which may be neutralized by cations such as sodium and potassium to form salts. Thus, amino acids react with alkalis to form salts. The sodium salt of glutamic acid, that is monosodium glutamate, also known as MSG, is usually commercially used as a flavoring agent. MSG is known in common parlance as Ajinomoto. Reaction with alcohols, that is esterification. With alcohols, proteins form corresponding esters, which are usually volatile. Reaction with amines. Amino acids in proteins react with amines to form amides. Now let us look at reactions involving the amino group. Reaction with mineral acids, that is salt formation. When free amino acids or proteins are treated with mineral acids such as hydrochloric acid, acid salts are formed. Reaction with formaldehyde. With formaldehyde, the hydroxymethyl derivatives are formed, which are insoluble in water and resistant to attack by microorganisms. Reaction with benzaldehyde. Proteins and amino acids form CIPS bases with benzaldehyde. Reaction with nitrous acid. This is also known as Barnes-like reaction. Amino acids and proteins react with nitrous acid to liberate nitrogen gas and produce the corresponding alpha hydroxy acids. The amino acids, that is proline and hydroxyproline, however, do not respond to these reactions. Reactions with acylating agents. This process is known as acylation. Acylation is brought about by many acid chlorides and acid anhydrides when amino acids and proteins react with them in alkaline medium. Reaction with FDNB, also known as Senger's reagent. In mildly alkaline solution, FDNB, that is 1-fluoro-2,4-dinitrobenzene, reacts with alpha amino acids to produce the yellow colored derivative DNB amino acid. Reaction with denyl chloride. The N-terminal amino acid of the protein may combine with 1-dimethyl-aminonaphthalene 5-sulfonin chloride, which is known as denyl chloride, to yield a fluorescent denyl derivative. Now let us look at reactions involving both carboxylic and amino groups. Reaction with triketohydrinidine hydrate. This is known as ninhydrin reaction. It's a very common reaction of proteins. Ninhydrin, that is indane 1,2,3 trione hydrate, is a powerful oxidizing agent, and upon its reaction with amino acids leads to oxidative decarboxylations of alpha amino acids, producing carbon dioxide, ammonia, and aldehyde with one less carbon atom than the parent amino acid. The reduced ninhydrin, that is hydrindantine, then reacts with the liberated ammonia and a mole of ninhydrin, forming a blue color complex known as Ruhemann's complex. Reaction with phenyl isocyanate. With phenyl isocyanate, hydantoic acid is formed, which in turn may be converted to hydantoin. Reaction with phenyl isothiocyanate. This is known as Edman reagent. Phenyl isothiocyanate reacts with amino acids to produce thiohydantoic acid. The latter, on treatment with acids and non-hydrolytic acids, cyclizes to form thiohydantoin. Reaction with phosgene. With phosgene, N-carboxy anhydride is formed. Reaction with carbon disulfide. With carbon disulfide, 2-thiopypthiazolidone is produced. Now let us look at reactions involving the R group or side chains present in protein. Burette test. Compounds containing peptide bonds produce a characteristic purple color when treated with an alkaline 0.2% copper sulfate solution. This is also known as burette reagent. This reaction is named burette reaction as it is also given by the compound called burette. The color deepens as the number of peptide bonds is increased, and the proteins produce a deep blue-violet color due to the possible formation of a coordination complex. All proteins and peptides except dipeptides respond to this reaction. Therefore, this reaction is widely used both as a qualitative test for the detection of proteins and for quantitative estimation of protein concentrations. Xanthoprotic test. On boiling proteins with concentrated nitric acid, yellow color develops due to the presence of benzene rings. This reaction is due to the nitration of the phenyl rings, which is present in the amino acids such as tyrosine, phenylalanine, and tryptophan, to yield yellow substitution products which turns orange upon addition of alkali. Millen's test. A red color develops when proteins are heated with mercuric nitrate in nitrous acid. This reaction is specific for tyrosine and takes place between mercuric and mercurous nitrates and tyrosine residues of the protein. Tryptophan also responds to this reaction to some extent. Hopkins-Cole test, also known as glyoxylic acid test. A violet ring develops upon addition of concentrated sulfuric acid at the junction of protein and glyoxylic acid solutions. This test is specific for tryptophan. Pauline's test. A protein solution develops blue color with phosphomolybdotungstic acid in alkaline solution. This reaction is due to the presence of phenolic groups in many proteins and is usually specific for tyrosine. Sakaguchi test. A red color develops when proteins react with alpha-nephthol and sodium hypochlorite. This test is used for the detection of arginine in proteins. Poly test. A protein solution develops red color with diazotase sulfonilic acid in alkaline solution. This reaction is specific for tyrosine and histidine. Ehrlich's test. With para-dimethylaminobenzylaldehyde in 12 normal hydrochloric acid, tryptophan develops a blue color. This test is known as Ehrlich's test. Now let us look at reactions involving sulfhydryl groups present in proteins. Nitroprusside test. Proteins develop red color with sodium nitroprusside in dilute ammonium hydroxide. This reaction is specific for cysteine. Sullivan test. Cysteine develops red color in presence of sodium 1,2-neptoquinone 4-sulfonate and sodium hydrosulfide. Color reactions of various amino acids present in proteins are shown in the accompanying table. Now, let us look at the mechanical properties of proteins. Certain specialized motor proteins or contractile proteins exhibit unique mechanical properties. Motor proteins may be classified into actin-based motor proteins. Myosin is an example of this class. It moves along actin filaments. Tubulin-based motor proteins. Tubulin is the building block of microtubules. Kinesin and dynein are tubulin-based motor proteins. They move along microtubules. Nucleic acid-based motor proteins. Examples include DNA polymerase and RNA polymerase, which move along DNA and produce force. The motor proteins have certain common mechanical properties. That is, they move along protein filaments. They can bind to specific filament types. They hydrolyze ATP and use the liberated energy for generating motion and force. Now, let us discuss the biological properties of proteins. Some of the biological or functional properties of proteins, especially food proteins, are as highlighted here. Solubility. That is, proteins' solubility promotes absorption and digestion of foods. Gelling capacity. Gelling property of food proteins enhance food. Water holding capacity. This promotes absorption and digestion of foods. Foaming ability. Foaming ability of certain food proteins enhance the palatability of foods. Adhesive property increases the stickiness of foods, whereas viscosity is a property which allow certain proteins, food proteins especially, to act as thickening agents in certain types of foods. In this lecture, we have had a thorough coverage of the major physical, chemical, mechanical, and biological properties of proteins. We hope that you have had a rewarding learning experience. We wish you a grand success in your further academic endeavor.
Behavior means an act which an organism does. It is a trait of any living entity. The study of behavior is called behavioral biology, or more precisely called ethology, a separate branch of biology. It is very interesting to study and know this because behavioral biology is very close to psychology, where human behavior in particular is considered. It has wide applications in forensic medicine and health. Ever since the evolution of man, he has been a keen observer and then an analyzer. This behavior of his has led to separate study of biology called ethology. The basic approaches to study of behavior are at ecological, that is interaction of an organism with its environment, and then ethological, that is whether innate, stereotyped, or genetic, chiefly predominant in lower organisms, of course a basic in higher forms too, and secondly, learned as is the case with higher forms of life. Introduction. Darwin's and his predecessors' natural history approach gradually evolved into the twin sciences of animal ecology, the study of interaction between an animal and its environment, and ethology, the behavioral biology. Ethology dates back to the late 19th and early 20th centuries, when scientists from various countries began exploring the behaviors of selected vertebrate species, example dogs by the Russian physiologist Ivan Pavlov, rodents by American psychologists John B. Watson, Edward Tolman, and Carl Leschly, birds by American psychologist B.F. Skinner, primates by German-American psychologist Wolfgang Kohler, American psychologist Robert Yerkes, etc. These studies were carried out in the laboratories in the case of dogs, rodents, and pigeons, or in artificial colonies and laboratories in the case of primates. These studies were oriented towards psychological and physiological questions rather than ecological or evolutionary ones. However, it was not until the 1930s that field naturalists such as English biologist Julian Huxley, Austrian zoologist Konrad Lorenz, and Dutch-born British zoologist and ethologist Nicolaas Tinbergen, studying birds, Australian zoologist Carl von Frisch, and American entomologist William Morton Wheeler, examining insects, gained prominence and returned to broad biological studies of animal behavior. These individuals, the founders of ethology, had direct experience with the richness of behavioral repertory of animals living in their natural surroundings. Their return to nature approach was a large extent a reaction against the tendency prevalent among psychologists to study just a few behavioral phenomena observed in a handful of species that were kept in impoverished laboratory environments. Approaches to study behavior. The chief aim of the psychologists was to formulate behavioral hypotheses that led to have general applications, example about learning as a single all-purpose phenomenon. Later on, this could be used as a deductive approach by testing the hypothesis through experimentation on captive animals. In contrast, the ethologists advocated an inductive approach, one that begins with observing and describing what animals do and then proceeds to address a general question. Why do these animals behave as they do? By this, they meant how do specific behaviors of these animals led to differential reproduction. Since its birth in the 1930s, the ethological approach, which stresses the direct observation of a broad array of animal species in nature, embraces the vast variety of behaviors found in the animal kingdom and commits to investigate behavior from a broad biological perspective, which has proved to be highly effective. One of Tinbergen's most important contributions to the study of animal behavior was to emphasize that ecology is like any other branch of biology, where a comprehensive study of any behavior must address four categories of questions, today which are called levels of analysis, namely, number 1, causation, number 2, ontogeny, number 3, function, and number 4, evolutionary history. Although each of these four approaches require a different kind of scientific investigation, all contribute to the solving the enduring puzzle of how and why animals, including human beings, behave as they do. A familiar example of animals' behavior is a dog wagging its tail, which serves to illustrate the levels of analysis framework. For example, when a dog senses